Welcome to a special 15-year anniversary episode of Stories from the NNI. I'm Lisa Friedersdorf, Director of the National Nanotechnology Coordination Office, or NNCO. Today I'm pleased to welcome Michelle Bradbury, Professor of Radiology at both the Gerstner Sloan Kettering Graduate School of Biomedical Sciences at Memorial Sloan Kettering, or MSK, and the Weill Medical College of Cornell University. Michelle also serves as co-director of the MSK Cornell Center for Translation of Cancer Nanomedicines. She is the Director of Interoperative Imaging in the Department of Radiology and co-chair of the Innovations and Technology team at MSK. Michelle, thank you so much for joining us today. Before we get started, could you just give us a little bit of information about yourself and how you got involved in nanotechnology?
Sure. My background is very diverse, but largely has gone from a bachelor's in chemistry into the biomedical engineering areas as part of my graduate training, which had a medical imaging component. So that led to my medical training at George Washington University School of Medicine. And this combination of physician scientist has really enabled me to move within a number of disciplines to identify gaps, for example, in some of the what used to be called contrast agents more broadly, but we call them probes now on the nano side. But it enabled me to really work in an area of medical imaging, probe developments to address limitations in clinical applications that I heard from many colleagues, largely my surgical colleagues. And in addition to that, the nanotechnology piece, it became evident that many of the probes we had were just not very specific. They've been around for decades, but there were a number of key limitations. And I reached out to my collaborator, Professor Uli Wiesner at Cornell University. He's been my collaborator for 13 years now. He had been working with a number of different particles. I saw the potential to develop more specific probes for targeting cancer, for example. And I had come here to Memorial Sloan Kettering in 2003. So basically uniting the various parts of my background and moving it into the nanotechnology realm in the field of medicine led to a lot of development that's happened to date during my 16 years here.
Well, as you know, the National Nanotechnology Initiative is celebrating its 15-year anniversary. And during this celebration, we're reflecting on the progress that has been made. From your perspective, can you discuss some of the key research advances that you think have been made in nanomedicine over the past 15 years?
Sure. The area of nanotechnology tied into medicine, that's been going on for 20 plus years, but I think there have been major advances along both the development of probes that are both directed at imaging disease as well as treating disease in various settings. These advances in the number of products that have been put forth in nanotechnology, it speaks for itself when you look at the explosion of probes for all kinds of applications and the ability to really design the surface chemistry better to address certain types of biological and clinical questions. I think also the nature of the imaging capabilities that these platforms can achieve has also really expanded so that you can use a single product for PET imaging, MRI, optical. So you could gather information that's complementary to the question being addressed. Our characterization tools, how do we look at the products in terms of their purity, the yields of our products, whether they are stable. A number of high-resolution characterization tools have emerged to better characterize the product prior to using it in clinical care. And I think, in general, there's been a much greater degree of understanding of the obstacles that have limited product translation over the decades. Moving from liposomes and more lipid-like materials and micelles to more engineered type products that really are diversified to address a range of clinical issues. The translational approaches have improved in many centers. The designs of the studies needed to advance products from bench to bedside has become much more sophisticated. There are multidisciplinary teams of people working, scientists and clinicians and regulatory groups working together to advance a more sophisticated and more, basically the probes have become very well studied across, I would say, radiology, depending on the application, surgery, medicine, pathology, many indications from the diversity of products that have emerged. So I think since I've been here at Memorial Sloan Kettering in 2003, and even earlier, there's been a really large development of all of these technologies that have been used either as single agents or with devices like camera systems and other imaging devices to do a much better job improving our sensitivity, specificity, and resolution for detecting disease.
Your bio talks about your collaboration in the co-development of nanoparticles called CDOTs. Can you talk a little bit about CDOTs and how they're being used?
So CDOTs that are aptly named after Cornell dots were developed by my collaborator, Professor Uli Wiesner. When I met him, he had already begun to size the particle down from something that's about 30 nanometers or more traditional nanoparticle size to sizes below 10 nanometers, which would then facilitate renal clearance. So this was a very significant advance for the field at the time because these ultra small products were, there were quantum dots, I guess, that were also available as semiconductor materials that there were large efforts to improve the toxicity profiles on, but then there weren't very many other, there may be some iron oxide agents that were also ultra small or gold. But I think that there was really a paucity of probes in this ultra small category that could be cleared out of the body so that you could really improve your identification of lesions in the major organs. So in other words, it's not going to the liver, which would obscure the ability to see metastatic disease there, for example. So by being below the renal threshold for clearance, major advance was achieved such that we could not just get it out of the body, but also functionalize the surface to target tumors. So it became a target or clear platform. And I think that actually led to a lot of preclinical studies that showed that this was a promising platform for clinical use. We looked at a range of biological properties ranging from specificity, binding affinity, potency, pharmacokinetics, because we adapted the particle for imaging and we could do PET imaging. And the particle also has a fluorescent dye in the core to be able to monitor, say it gets into the tumor, you could look at the tissue sections and see how it penetrated and distributed by the optical signal from the particle when you shine light on it. So I think the ability to develop the capabilities of such a platform and improve the pharmacokinetics, which is what we saw, led to the translation of that product. So a lot of work was done between our teams. It was a real team approach to improve the properties of such a material to make it ready-made for patient care.
We hear a lot that the NNI really brought people together across a wide variety of disciplines to work together, and that it really created this ecosystem where people were working maybe a little bit outside their comfort zone with people of an entirely different technical background. Is that what you've experienced? And from your perspective, is that something that has enabled advancements to occur more quickly?
Absolutely. I think that this has even been the results of collaborations such as mine with Cornell really led to, I think, a lot of properties being optimized in such a manner that it really facilitated the translation of the product. So you need the engineering group, the group that does polymer chemistry or just chemistry of materials, and also does a lot of characterization with HPLC and other approaches, understands how to functionalize the surface of the product so that in tandem with us, we can really go back and forth and troubleshoot until we get the most optimum product. Because on their side, for example, they know all about, if you have a fluorescent nanoparticle like a CDOT, the brightness of that CDOT was much greater than if the dye was appended to the outside. I mean, that was a serendipitous finding by Professor Wiesner's group. And this led to a number of indications. We could have, on our end, we would have not been working on that piece, but we worked to help improve the surface chemistry by testing, by creating an imaging agent, by putting a PET label on and doing screening pharmacokinetic studies to understand, is this the behavior that would be favorable for clinical translation? And also, does the targeting, depending on the number of tumor models we work with, is the target to background ratio, for example, how much accumulates in the tumor versus off-target sites, is this appropriate to move forward? Is this going to give very good contrast to enable us to look at metastatic disease or to use in the operating room, given the optical component with a camera system? Is this sufficiently bright? So I think there are a lot of questions that need the engineering group and the group that does chemistry to really work hand in hand with us, which speeds the process.
So you've discussed a number of applications and the advances that have enabled early use of some of these materials. Looking to the future, what do you think are some of the remaining challenges that require, additional research and development? And what particular challenges do you think nanotechnology will help clinicians such as yourself solve?
It's a great question. And I think there have been many talks around challenges to the translation of nanomaterials. And, these materials are more complex, but they also offer a lot of opportunity to address properties that have not really been optimized well, biological properties, in vivo properties. We can actually tune the surface chemistry. We can make adjustments to the surface chemistry of the product to keep improving the biological properties that we're looking at. And I think that is really significant. We can look at several biological properties at the same time. We can not only image and make sure that the platform goes to the right places, but we can look at the uptake in tumors, its penetration and distribution. Is it the best platform to deliver a drug, for example? We can look at all of these at the same time because we're working with a platform, and we can modify it as an imaging agent. And so we can use different imaging modalities to pick out or answer different questions related to the behavior of the platform in vivo. But some of the challenges to translation, which have related to having an optimized surface chemistry that's reproducible and stable in the body, I think we've come to understand what is likely to work from a lot of experience from across many different platforms out there. What are the limitations of different platforms? Where can some platforms offer advantages? What are the best ways to move these through the translational apparatus to the FDA? I think that other challenges, actually moving from a clinical trial into a commercialization area, where sometimes we're doing trials and we want to see what are the best products to take forward that could really help a lot of patients. And sometimes not every product is optimal for that, but I think that there are challenges when you're going from one stage to another to make the best decisions with a group of people that do this all the time, the people that help to commercialize platforms for patient care. And they work with the clinicians and scientists to say which of these has the most promise for what can be achieved.
Michelle, I think based on our discussion today, I agree. It's a very exciting time, and I want to thank you for taking the time to talk with us today. Do you have any closing thoughts that you'd like to share with our listeners?
I think it's really important for the nano field to train the next generation of individuals that can really just pick up the mantle from us and continue to move forward. The education and training piece is extremely important because of the complexity of this area and because of the enormous potential that can be realized. We spend a lot of time training students, many individuals from the high school level and beyond, and we just encourage anyone who's interested to contact us and help to develop these products, the next generation products, which will improve our care of cancer patients. And again, I also think that the multidisciplinary teams that are working together in the best way possible now is a significant advance for the field and will continue on for decades to come. I think that it's great to bring many groups together that can help solve the problems for patient care by working together that have really made it difficult in the past. But we have a number of new technologies and tools, and we have many experts now, at our respective hospitals and outside, as well as the business arm that can help to make decisions as to what to advance that could be best used for many patients. So I think we need to continue to keep up all of that good work. And I think we'll see some very beneficial results from these types of products. And we'll continue to see this into the future.
Thank you for joining us today for this special 15-year anniversary edition of Stories from the NNI. If you would like to learn more about nanotechnology, please visit nano.gov or email us at info@nnco.nano.gov. And check back here for more stories.